Okay, so now I want to talk about proteins and then nucleic acids. So in proteins, as with all of these macromolecules, you have carbon, hydrogen, and oxygen. We also have some nitrogen and some sulfur. And some examples: hemoglobin, which is a protein found in your blood that helps transport oxygen. Collagen, which is a structural protein found throughout your body that helps sort of hold things together. I believe we mentioned in class keratin, which is also a structural protein that is part of your hair and fingernails. But there are proteins throughout your body that do many different things: transport, in and out of cells, moving things around cells, support, part of your immune system as well, certain hormones in your body, many different functions. The basic unit of a protein is the amino acid, and it consists of the amine group, a carboxyl group, thus amino acid, because you remember the carboxyl group can sometimes lose that hydrogen and acidify a solution. And then there's this carbon, which has a hydrogen attached. And so that is the basic structure of an amino acid. And down here you have what's called the R group, or sometimes known as the variable group, because when we look at our 20 amino acids, we see that here's glycine, here's the amine group, carboxyl group, the other hydrogen, here's our variable group, it's just a simple hydrogen. Alanine, there's our variable group. Valine, there's our variable group. You can see some of them, tryptophan, get quite complicated with these double rings and such. Again, 20 different kinds. Some tend to act as bases. Some are polar and have a charge. Some tend to act more as acids. But again, they all have that basic structure. And notice here that this is showing that when they're in solution, the amine group will sometimes grab onto an extra hydrogen, and the carboxyl group will tend to lose a hydrogen, and so they can have charges because of that. Alright, now, so to build a protein, you have to put your amino acids together. You have to get your monomers and form a polymer from that. Just like with other things, we put them together using a dehydration synthesis reaction, and we form what's called a peptide bond. And so when you put a bunch of amino acids together in a long strand, thus you end up with a polypeptide, that sequence of amino acids there. That's sort of the first level or primary level of organization of proteins, but then all proteins will form hydrogen bonds in certain places, forming these corkscrews called an alpha helix, or these sort of folded accordion-like sheets that are known as beta-pleated sheets. So some portions of the amino acid sequence will do that. So that's the secondary level of structure, and then they will fold up into this three-dimensional blob-like structure, and that's the tertiary or the third level of organization, again, that all proteins have. Some proteins will then go to the next level, the fourth level or quaternary level of structure, in which you get separate subunits that bond together, making a really big protein. And this one, I believe, is supposed to represent hemoglobin, because hemoglobin consists of 4 separate subunits. Okay, now nucleic acids. So carbon, hydrogen, oxygen, also a lot of nitrogen and a fair amount of phosphorus, but no sulfur. Our examples: RNA or ribonucleic acid, DNA or deoxyribonucleic acid. The monomers of nucleic acids, they're known as the nucleotides. And here's your basic nucleotide. You have what's called the nitrogen base, you have a sugar, and a phosphate group. And so to take a single nucleotide and make up your nucleic acid, you have to put a bunch of them together. And notice, see, here's a nucleotide, here's another nucleotide, etc., consisting of this sugar, nitrogen base, sugar, and phosphate. And notice that they're bonded. One sugar is bonded to the phosphate of the next one, which its sugar is bonded to the phosphate of the next one, etc. And the sugar-phosphate bonds sort of form the side, or what's sometimes described as the backbone of the molecule. And the nitrogen bases are sticking off to the side here. Alright, so we have 5 different kinds of nitrogen bases here. We have one that's what's called cytosine, and this is on page 50 in your book. Thymine and uracil, and then we also have adenine and guanine. So notice we've got 3 that have the single ring and 2 that have a double ring. The single ring ones are known as the pyrimidines, and I like to remember them as picut. Pyrimidines are cytosine, uracil, and thymine. These guys are known as the purines, and the purines are adenine and guanine, POG. Alright, now the sugar in DNA and RNA is different. So in DNA we have the deoxyribose sugar, deoxyribose, and in RNA we just have the ribose sugar. You can see they're similar, but there's a slight difference. There's this extra oxygen down here in the ribose sugar. Alright, so we're going to have a whole chapter on DNA, and we'll talk more about the structure and how RNA is a single-stranded molecule, just a single strand of nucleotides, but that DNA is a double-stranded molecule. And they have different jobs within the cell. Now the book, the end of this chapter, I don't have a picture, but they also talk about ATP on page 52, which is basically one of these nucleotides, but it has 3 phosphate groups, ATP, instead of 1. And we'll see that, or the book talks about how ATP is basically the energy currency in the body, and when we study cellular respiration and photosynthesis, we'll see the important role that ATP plays in those reactions and then how cells make use of it. Okay, thanks.